Our next speaker is Dr. Elise Dennis. She is the Vice President of Roche Tissue for Global and Medical Affairs, and she works all around the world, and she works tirelessly, and she's an amazing person. So I'm really glad that she's here with us. So ladies and gentlemen, Dr. Elise Dennis.
Thank you again, Amanda, for that very kind introduction. Thank you to you and to Gina for this amazing opportunity to be part of this summit. This is such a special event. And really, I'm so inspired by everyone that I've met, everyone that I've worked with, everyone that I met yesterday. It really feels like coming to a family reunion. And it has that sense of everyone just loves each other and everyone just supports each other. And it's truly an honor for me to be here with my colleague, Amy Hanlon-Newell. So thank you for welcoming us so warmly. Amanda does have a very special place in my heart. And as I've gotten to know her and her husband, Gary, it's just been really remarkable. And she's taken me back to where I started my career, which was as a physician dealing with patients. And so I was a physician back in Africa, in Zimbabwe and South Africa. And mostly what I did was treat patients who had hematologic malignancies, so leukemias and lymphomas, young children, young adults, older adults. And this was back in the late 1980s and early 1990s, when cancer treatment was not like it is today. Back in those days, we had different combinations of chemotherapy, we had radiation, and we had surgery. And that was it. We had very little in the way of guidelines. There was almost no support networks for patients. But yet, we always had hope. We always had optimism. We always knew that when people were doing research, that things were moving. Sometimes it felt really slow, but we were making progress. At the same time, back in Africa in the late 80s and the early 90s, HIV and AIDS was huge. And no matter what branch of medicine you were in, that touched all of our lives. And at that point, a diagnosis of HIV was before we had any antiretroviral therapies whatsoever. So a diagnosis of HIV was a certain death sentence, absolutely. And in my career lifetime, and I still think I have a long runway ahead of me, I hope, right? But in my career lifetime, I've seen treatments for HIV and AIDS. I've seen AIDS move from being a fatal disease to a chronic disease. And I've seen tremendous progress in the field of cancer research. There has never been a more exciting time to be in this field of cancer research. Now, I left clinical practice a while back because I was frustrated. I did not have effective therapies for my patients. And there is nothing worse when you are a physician and you are sitting in front of a patient and you have nothing to prescribe. And I wanted to be part of a movement where I could translate that science and bring new treatments to patients. And that's what I've done. I joined the biopharmaceutical industry and got involved with clinical trials right from the very beginning. And it really has been an immense privilege to get to work with the world's leading scientists and oncologists and pathologists to really uncover this world of science. And my hope is that we're going to completely rewrite those medical textbooks. In fact, we're going to throw out those textbooks. By the time a textbook gets printed, it's out of date. So we want to make sure that we can take new therapies, bring them, because when Gina was going through all of the patients that we have lost, that is just unacceptable. Even though we've made progress, we are a long way from being able to say that every patient who is diagnosed with cancer will be a survivor. But there is tremendous love in this room. There is tremendous passion, commitment, resilience, tenacity. In fact, it's just unshakable stubbornness, right? We are going to get there together, okay? So thank you for having me. So my talk today is going to be about the journey of how we go from science to medical breakthroughs for ALK positive patients. But what I really want to do is to do a deep dive. I want us to really know cancer, really know ALK, all right? Knowledge is power, and knowledge is a force multiplier for change. It's knowledge that's going to take that snowflake that Gina talked about and turn it into an avalanche for us to change. So that's the intent of today. These are my disclaimers. I do work for Roche Tissue Diagnostics, Ventana Medical Systems, but the opinions and views that you'll hear today are entirely my own and don't represent anyone else. Okay, so here's what I'm going to cover in the next 40, 45 minutes. We're going to start off with really understanding the basics of cancer. Now, I have to tell you that this audience is a tough audience because all of you have different levels of scientific understanding. Some of you are scientists and physicians in your own right, right? Some of you are not scientists or physicians, but you know way more than your own physicians. And some of you, it's kind of overwhelming, all right? So what I wanted to do is really make sure that we are all on the same page. So we're going to start off with some real basics. So for those of you that are scientific gurus, just bear with me, okay? Because we'll get there. But what I don't want is for anybody to leave today to say, oh my gosh, that presentation just flew on top of my head and it was so boring and I fell asleep. So that's what I don't want. I want all of us to start off on the same page and we're going to build it up. So we'll go from the science of cancer, the basics, and then we'll talk about the journey of ALK, from science to medicine, and then we're going to talk about the importance of biomarker testing. Okay, everyone with me? Everyone awake? All right, so I hope that, do we need to bring the light a bit down, Ryan? So I do have some images in here of lung cancer. So if any of you are a little squeamish, just turn your eyes down. But from everyone I've spoken to, I think you'll be okay. But I just wanted to give you that heads up ahead of time. Okay, a little louder. Yeah. Ryan, can you bring up the volume?
Bring it back. If I bring it up a little bit, it's going to start feeding back. If you could just speak like you're hitting the back of the room there.
Okay. Actually, I'm going to take singing lessons next time so that I can, but keep me on track because the one thing is I can talk soft and then I can talk really quickly. So if anything that happens, just wave and I'll slow down. Okay. All right, let's start with the science of cancer. Each one of us is different. Each one of us is beautiful in our own ways, and each one of us is unique. But what we have in common is that we are all made of cells. In fact, 40 trillion of them. There's actually a paper that estimated the number of cells in the human body. If anybody's interested, I can share that with you. But fair to say that we have a lot of cells in our body, and our cells are constantly dividing. So in fact, that is the most important concept for us to grasp if we really want to understand what cancer is and what causes cancer. Okay, so if we think about normal cell division, all right, every single cell in our body has exactly the same genetic blueprint. It has exactly the same DNA. And so when we have a mother cell that divides into two daughter cells, the most important concern about cell division is making sure that we can maintain that genetic blueprint. So we want to make sure that each of those daughter cells has the identical blueprint. And as those cells divide and create their own daughter cells, that genetic blueprint is going to be the same, okay? So that's the intent of cell division. Now, the intrinsic machinery that governs DNA replication, so replication of our genetic material and cell division, it's very complex and it's not entirely perfect. So every now and again we'll have a cell where we have a genetic alteration that is introduced. Now we also have different sets of machinery that can actually detect any errors in that replication process and can actually correct those errors. And if that happens, that's great. But sometimes that machinery isn't perfect either, in which case we cannot fix that cell, and so we have another set of machinery that actually will take care of that cell by eliminating that cell. And it kills that cell through a process called apoptosis. And so another way of thinking of apoptosis is programmed cell death. And I've heard some people refer to this as civilized cell death. So it's actually a very normal process. It's a way that cells can actually say, you know, it's time to actually kill the cell without creating too much of a mess. And so this is a perfectly normal process. But what happens when we have cancer is that these processes break down. And we end up with these abnormal cells that have these genetic alterations that continue to grow and continue to divide uncontrollably. So that's what happens when we have cancer. So we can really think of cancer as a genomic disease. Now, when I say that, often people will say to me, well, does that mean that my cancer is inherited if it has to do with my genetics? And that's not the case. Only about 5% of all cancers fall into this category that we think of as being hereditary cancers. The rest of all the other cancers are actually the result of an accumulation of genetic mutations over time. And so for us to understand what these genetic mutations are, it's really important that we understand what the normal blueprint of the cell looks like. So our genetic code is housed within our DNA. And our DNA is contained within chromosomes. So really a chromosome is really a long molecule of DNA that's really coiled up. And those chromosomes are housed within our cell nucleus. Now, the DNA is actually formed by two strands that twist along each other to form a double helix. And those strands are formed by sugar and phosphate molecules that are bound by what we call bases or nucleotides. So these are the building blocks of DNA. And there are 4 building blocks: adenine, thymine, cytosine, and guanine. So just remember it as ATCG, all right? So DNA, double helix, these building blocks. Now, these base pairs, these building blocks, can be ordered in any sequence, but the specific sequence that they appear in is what codes for making proteins. So along the chromosomes, these coding sections of the DNA, we call them genes. And along the chromosome, those genes have a specific location, all right? So when we think about a cell that's going to divide, before that cell divides, it has to make an identical copy of its DNA so that it can pass 2 sets of DNA to the daughter cells. And that process is called replication. Now, when we want to translate DNA to make proteins, we don't directly make proteins from the DNA. The DNA is in the nucleus. What we do is we make a temporary copy of something called messenger RNA. It's like a short segment that mirrors that DNA, and that moves from the nucleus to the cytoplasm of the cells. And we have these protein-making machines in the cytoplasm called ribosomes. And that's where the messenger RNA will bind and make those proteins, okay? And that process is called transcription to make the messenger RNA and translation to make proteins. So it's a fairly complex set of processes going on, but the most important thing to remember is that it's happening in 40 trillion cells, okay? And each cell has the whole genome of 3 million base pairs. So that's 6 million bases that we have to replicate each time we want to make a new cell. So even if I factor in like maybe a 0.1% error, because nothing's perfect, there's still going to be 6,000 bases I'm going to get wrong, right? So things aren't perfect, and things can go wrong. And things can go wrong just by chance, right? Okay, so let's focus in more on the chromosomes. So this is what we call a karyotype. Now, we can actually isolate chromosomes and stain them and put them on a slide and take a picture. And then with that picture, we can cut out those chromosomes and lay them next to each other to make this composite image of what we call a karyotype. And we have people who are trained as cytogeneticists who can actually look at these karyotypes and look at these chromosomes and see if there are any abnormalities. Now, normally, in pretty much all of our cells, with the exception of sperm cells, egg cells, and red blood cells, every other cell has got 23 pairs of chromosomes. So that's 46 chromosomes. And they're laid out in pairs. It's a bit like Noah's Ark, right? The animals go in 2 by 2, so we have the chromosomes 2 by 2. So this is what our chromosomes look like normally. Now, I've just said that cancer is a disease where we have genomic alterations. So we can anticipate that in cancers, we might see quite a different picture. And here is a picture of a patient who has colon cancer. So I think you can see, even from the back of the room, that those two pictures really look quite different. So what we can see here is like chromosome 1, there's 3 chromosomes over there. Chromosome 7, there's 4 chromosomes. Chromosome 5 only has 1 chromosome. So actually, back in the early 1900s, in 1902, a German biologist called Theodor Boveri actually had a hypothesis that cancer was a disease caused by scrambled chromosomes. And it was really quite a remarkable hypothesis because back in those days, he had no technology to even prove that, right? But he was a really smart guy. That was his hypothesis. And it was only in 1960 when the first chromosomal abnormality associated with the cancer was actually identified. And that was the BCR-ABL gene in chronic myeloid leukemia. So actually, this is really something that, this is a picture tells 1,000 words. We can see that these are these genomic abnormalities that are associated with cancer. And since then, we have associated 1,000 small chromosomal abnormalities with different cancers. Okay, so now we have our DNA, and we have to decipher our genome, right? And this can be quite a daunting process, because at the end of the day, what we're trying to do when we sequence our genome is identify all of those A, T, C's, and G's in a different order, right? But it's really quite complicated. However, if we think about information in the digital age, right, the reason why you're seeing this picture on the screen, the reason why you see pictures on the TV, that you hear songs on iTunes, is because we can take zeros and ones and we can translate them, okay, into pictures, into sound, into our digital information. So it's kind of the same principle, all right, except that with DNA we have 4 letters instead of 2 numbers, the zeros and the ones. All right, so let me give you a very, very simple illustration of how sequencing works. So we're going to take a series of letters here. This is what happens with our sequencing. Here we see a series of garbled letters, and we have no idea what they mean, right? It's completely nonsense. So the next step after we sequence our genome is we really have to analyze that genome. So in this illustration, I've taken those letters, and now they read, "The boy is in the tree." But now I have to interpret that information. Okay, so what does that mean? Well, he might be stuck. And then once I've interpreted that information, I have to take an action. All right, so what's my action? All right, I may need to get a ladder. Okay, now that's very, very simple, but that just gives you the concept of what happens when we sequence information, we analyze it, we interpret it, and we have to take an action. Now, what are some of the things that can go wrong with the genome? So let's think of the boy being in the tree. Well, we can have things called single base substitutions. So now in this case, I've substituted T for B, and it's become the toy is in the tree. So now my level of anxiety is not quite as great. I'm going to have a different action because while the boy's in the tree, I might go run get the ladder. If it's his teddy bear stuck in the tree, I might just say, you know what, it's just going to stay there. So my action's going to be different based on these single base substitutions. All right, the next one is insertions. So now instead of the boy, I have the body in the tree. And in this case, I'm not getting up the ladder. I'm going to call the police. So it's a completely different action based on this mutation, all right? All right, now we have a deletion. So now I have the oi is in the tree. And that makes no sense to me whatsoever. So I'm not worried. I'm going to completely ignore it because this is missense, okay? Then I can have amplifications. The boy's in the tree, the boy's in the tree, the boy's in the tree. I am going to pay attention because it is screaming loud in my head. So that's a different action. And then we can have things called translocations. And we can also think of these as fusions. So in this case, instead of the boy being in the tree is in the boy. So that's a whole different level of anxiety and a very different action, okay? So these are the things that can go wrong with our genome. And it really is that straightforward. So let's apply this knowledge to some real-world examples. But before we get there, you're also going to hear a lot about driver mutations and passenger mutations, all right? Driver mutations are those changes that we see in that genome that actually do as it says. It drives the cancer. It's what makes a normal cell become a cancer cell and stay a cancer cell, right? It's what drives that uncontrolled growth, that uncontrolled replication and proliferation of those cancers. Those are drivers. Now, drivers give cancer cells their strength. That's why they survive. But they are also the Achilles heel of a cancer. Because if we can find what that driver is and we can find a target to block that driver, then we can kill those cancer cells. And cancer cells get addicted to these drivers. It's something we call oncogene addiction, right? So the key then is to say, what is my cancer cell addicted to, and how do I block that so that I can kill that cancer? And that's what we do when we have targeted therapy. Now, the other types of mutations you'll hear about are passenger mutations. We have a lot of random mutations that are really incidental. They don't appear to have any impact on your cancer. Now, that's not to say that maybe sometime in the future we may say, well, here's a role that we can identify, but at this point, they don't seem to make any difference. So we call them passenger mutations. They're just along for the ride, but they're not doing anything. Okay, now let's apply this example to some more biological types of examples. So here we have a chromosome, and here we have a gene of interest. So here we have a base substitution. Here we have insertions and deletions. And here we have amplification. And here we have those translocations. So what happens in translocations is that a gene from one chromosome kind of fuses with a gene from another chromosome. And I really want you to pay attention to that because that's exactly what happens in ALK Positive Lung Cancer. Okay? All right. The key message here really is that cancer can strike anyone. All right? Anyone who has cells who are dividing, and if those cells are dividing, we're introducing genetic alterations, okay? So those cells are dividing, we have this initiating mutation, those cells then acquire more abnormalities, they become unstable, they then acquire these hallmarks of cancer, and so that cancer grows, right? But if you really think of it, the analogy I like to use is really that of a combination lock, all right? So if you think about a combination lock that's spinning, right? If you hit just random numbers, nothing's going to happen. You're not going to open up that lock. And you know that if you want to open a lock, you have to get the right numbers in the right sequence before that lock's going to open. So that lock can spin and spin and spin, and you can get random mutations and nothing will happen. But in order for a cancer cell for a cell to become a cancer cell, there really has to be a specific set of mutations in a specific order, right? So there's a lot of randomness around this collection of mutations that gets to a cell becoming a cancer cell. Now, one of the things that we have really learned a tremendous amount about in the last, I would say, decade, and especially in the last 5 years, is the role of the immune system in cancer. Now, we have recognized that the immune system, in fact, has a very important role to play in cancer, and it has a dual role to play in cancer. On the one hand, the immune system can actually control and eliminate cancer cells. But under certain circumstances, the immune system can actually be a facilitator of tumor growth and allow those cancer cells to continue to flourish. And there's a concept here called immune editing that I want to explain to you. And there's 3 phases of immune editing. The first phase is elimination. So here is when a cell develops those genetic abnormalities, our immune system can recognize that cell as no longer being a normal cell. It's now what we call non-self. It recognizes it as foreign. And remember, our immune systems are really developed to protect us from infections, so from bacteria and so viruses. So it's those same processes. When it sees something foreign, it's going to go into, hey, I need to get you out of my system because you don't belong here. So that's what happens when we can detect these cells as being foreign. We can mount an immune response, and we can eliminate those cancer cells. Now, if that happens, that's the end of that process, and we may never have known that we had any cells that were about to become cancerous in our body. That's the end of that. However, if our immune system is not able to take care of those cancer cells, the next phase is equilibrium. And so what happens here is that our immune system keeps those cancer cells in a state of functional dormancy, right? So they're kind of coexisting, but there's this tension between those immune cells trying to kill the cancer cells and those cancer cells trying to evade the immune system. And remember, this is a dynamic system, right? So they're constantly trying to outcompete the other. Now, if the immune system wins, then we go back to elimination. But that cancer cell, those cancer cells try really hard. They'll adapt. They'll take on new mutations, right? They'll develop tumor variants, so clonal variants, to develop cells that the immune system can no longer recognize and can no longer destroy. And when that happens, we end up in the escape phase, and then we develop cancer that is clinically detectable. So that's what happens with the immune system and cancer. And if you really think about it, if we go back to the principles of Darwinian evolution, right? That's why we evolved to be a successful species. That's how animals evolved to be successful animals, right? So the principles of variation and adaptation and selection and fitness for selection under certain pressures, those exact same principles apply to cancer cells. So cancer really is a very complex adaptive system. Okay? So that's the basics about cancer. Is everybody still with me? Yeah. Good. All right, so let's recap. Cancer can strike anyone, all right? Cancer is a genomic disease. It's caused by the accumulation of mutations over time. There are many different types of mutations. The immune system plays an important role in cancer, and cancer is a complex adaptive system. Okay, let's move on then to get into the ELK journey, the story from science to medicine. All right, so ELK in cancer, that's okay. ELK in cancer was first recognized and identified in lymphoma. Now, for those of you who were on the tour yesterday with the American Cancer Society, they talked about funding the research of a young investigator called Stephen Morris. And he was at the St. Jude's Children's Cancer Research Hospital. And so this is the work that the American Cancer Society actually sponsored with the young investigator. So he discovered the ALK gene in a type of lymphoma called anaplastic large cell non-Hodgkin lymphoma. And consequently, the ALK gene encodes for a protein called ALK, which is called anaplastic lymphoma kinase. Okay? So, now ALK, in fact, is normal. Each one of us has got an ALK gene. And normal ALK we call wild type ALK. And ALK is actually a gene that's expressed mostly in our nervous system. And expression is highest when we are neonates, because it plays a very important role in the development and the maintenance of our nervous system. So ALK is normal, ALK is healthy, and ALK in the right setting is exactly what we need, because remember, when we're born, our brains aren't really well developed. And so if it wasn't for ALK contributing to our brains developing, we wouldn't be sitting here as functional adults today. So ALK has a normal role to play. However, ALK is not found in normal lung tissue, all right? So it's found it's expressed in the nervous system tissue, okay? Now, what does normal ALK do, and what does normal ALK look like? So this is the normal ALK protein. It's a membrane-bound protein, okay? See the outside and the inside of the cell. Let me see if you can see here. Okay, it's the outside and the inside of the cell. Okay, now, when we have specific signals that say, you know, we think we need some growth here, the body ends up releasing what we call these growth factors. And in response to the growth factors, normal ALK proteins, two of them will come together. It's a process we call dimerization. And when those two ALK proteins come together, they actually stimulate a process called phosphorylation, which you can see happening over here, okay? And that phosphorylation activates a whole series of pathways downstream that really result in cell proliferation and cell survival. And that's really important if you're a baby and you're developing your brain, okay? So let's go back then to Stephen Morris and the paper that he published back in 1994 where they discovered the ALK gene in lymphoma. Well, that actually initiated a whole flurry of research because people said, well, if the ALK gene is implicated in lymphoma, are there other cancers where it may also play a role? But it took us more than a decade before a fusion gene in lung was identified. And it was the EML4 ALK fusion gene that was identified in a subset of patients with non-small cell lung cancer by a group in Japan. So this paper that came out was really a seminal paper and fundamentally changed the landscape of drug development for ALK inhibitors. So let's talk about this genomic abnormality that we see with ALK, these translocations that we see with ALK. So the ALK gene is found on chromosome 2. Now here we have two examples of translocations. The top example, you can see the ALK on chromosome 2 fuses with TFG, which is on chromosome 3. And in the bottom example, we can see the ALK on chromosome 2 fuses with KIF5B on chromosome 10. So these are translocations. Now these abnormalities are actually not very common. The most common abnormality that we see in ALK positive lung cancer is the EML4 ALK fusion gene. But this is a little different because it doesn't involve two chromosomes. It involves the same chromosome. In fact, it involves the same short arm of chromosome 2. And what we see with EML4 ALK is actually an inversion. So it's a really small and subtle change because the ALK gene is really close to the EML4 gene. But we see this inversion. Now, you might say to me, well, this is a really small change and a really subtle change. How can you tell me this really is true, this really exists? Well, I'm going to share with you some work that was done by a really brilliant scientist that we work with on our team called Dr. Hiro Nita. And what Hiro did is he actually made these probes where you can actually bind to sections of DNA. And he made a probe for the two ends of the ALK gene and the middle part of the ALK gene. And he then put a dye, this color, this pigment, a different one on each of these probes. So for the one end of the ALK gene, you could see it was pink. The middle end was blue. The middle was blue. And the gold, this end was gold, okay? So pink, blue, and gold for the ALK gene. And when he took cells that were ALK wild type, so these are cancer cells, but they're not positive for ALK, for the ALK fusion, if you look very carefully, you can see the color, pink, blue, and gold, all right? So that's the ALK gene, pink, blue, and gold. Now, when we have the translocation, what happens here is that part of that EML4 moves from here to bind to that end of the pink part of the ALK gene. And then the middle and the other end kind of flip. There's that inversion. So now the colors have changed. Now we see pink, gold, and blue. And you might say, well, can you really see that? And so here we have lung cancer that's ALK positive. And when we look, we can see pink, gold, and blue. So that's that inversion that we've seen. But it's a very small inversion. It's very subtle. But that's the most common abnormality that we see in patients with ALK positive lung cancer. Okay, so that ALK gene, that EML4 ALK gene, encodes for a different protein. It's no longer the wild type ALK protein. We now have EML4 ALK, which is a different protein. So let me show you how this protein behaves differently. So remind you, the normal ALK, the wild type ALK, remember, you have to have a signal to say, come together, dimerize, phosphorylation, activation. But that signal can be turned on, it can be turned off. So it's only active under appropriate circumstances. What happens when we have the abnormal ALK is that that ALK protein now becomes cytoplasmic. It's no longer on the surface. It no longer responds to signals. It does its own thing. It dimerizes on its own. It phosphorylates on its own, and it activates those signaling pathways on its own. Those signaling pathways are on all the time. And that's what's driving that uncontrolled growth and proliferation in those cancer cells. Okay, everyone with me? Good. All right, so how does this translate into, how do we take that science into drug development? So the history of the development of ALK inhibitors goes back to 2005. Now, for those of you that were still awake and were paying attention, you might remember that the paper that came out from the Japanese group was in 2007, right? So you might say, okay, well, how can you tell me the history started back in 2005? Well, it really did. It's actually a fascinating story. So back in 2005, Pfizer was looking at different compounds in lung cancer. And they screened a whole bunch of compounds to see what targets these different compounds would be able to block. And so one of these compounds actually blocked a target called Signet, which was known to be implicated in lung cancer. It just so happened that it also blocked a target called ALK. But back in 2005, no one was thinking of ALK in lung cancer. So they said, okay, we're going to develop this drug as a c-Met inhibitor in patients who have lung cancer. So they actually had their first patients that were dosed back in 2006. So those clinical trials had started. So in 2007, when EML4 ALK was identified in lung cancer, the Pfizer team said, wow, this is really interesting. This is really exciting. We have this compound that's actually a dual inhibitor for ALK and for c-Met. And what we're going to do is we think that this could really be impactful. We're going to pivot. We're going to switch from using this as a MET inhibitor to looking at this as an ALK inhibitor. But the problem was that they didn't have a test. Because remember, ALK positive composes about 4 to 5% of all non-small cell lung cancer patients. So you don't want to give an ALK inhibitor to every lung cancer patient. You only want to give it to those patients who are ALK positive. But we had no test to test for ALK back in those days. So what they did is that they approached a very smart physician and scientist at Mass Gen called Dr. John Iafredi. And they said to him, can you make us an ALK test? And so what he did is he created an ALK Fish Test. So FISH stands for fluorescent in situ hybridization. And he made this test in his lab. So it's what we call an LDT, a lab-developed test. So he could run this test in his lab, but what you can't do with a lab-developed test is take that test and distribute it to a bunch of different labs and run that same test. But they wanted to have a test really quickly to say, we want to test the proof of concept and see if this is going to work. So that's what they did. So the first ALK patient was enrolled back in 2007, the same year that paper came out, because they had these patients that they had already screened. And very early in 2008, they saw their first responses, which was so remarkable. And they said, you know what, we need to make this a much bigger trial. But to do that, they then had to have a test that could actually go to a larger number of labs. And so they partnered with Abbott, that's a diagnostic company, that actually took that FISH test but made it into a test that could then be taken to a number of different labs. So once they had that, they could have much bigger phase 3 trials. In 2010, their paper came out in the New England Journal of Medicine. Early in March of 2011, they submitted all of their data to the FDA. They had an accelerated review, and by August of that year, Xalkori was approved. And so at the same time, the test for ALK positive, the test from Abbott, was approved as the companion diagnostic. Now, this was completely unprecedented. You know, up till this point, drug development could take like 10 years before you could get a drug approved. But from the time we identified the science to having a drug in the hands of patients, it was 4 years. That was truly, truly remarkable. And that was really the result of so many people working together. It was patients, it was physicians, it was the FDA, it was the whole community coming together to say, we need this drug in the hands of patients. So how then do we make a diagnosis of lung cancer? Well, cancer patients rely on a multidisciplinary team. So as patients, your strongest relationship is going to be with your oncologist. That's the person that you're going to interact with the most. But your oncologist has got a whole team of people working to support him or her, to help make those right decisions. And to make decisions, we need a whole series of different tests. So you'll have radiologists, endoscopy, surgeons that are going to be looking to get samples of tissue to make those tests, and they then end up in the hands of the pathologist, who really is at the center of all of that diagnostic information that's used by your oncologist to make decisions. So many of you, I'm sure, know your oncologist very, very well, but I'm sure very few of you know who your pathologist is and really what the pathologist does. And pathologists are physicians, but they're also lab experts. They know about tests. They understand all the different tests that are out there. They know how to run those tests. They know how to interpret those tests. And they are an essential partner to the oncologist to say, how do I interpret this to make the right treatment decisions for my patient? So what I'm going to do now is really focus on how the pathologist looks at lung cancer. Okay, so to make a diagnosis of cancer, you have to start with tissue. Tissue is essential. No matter how high your index of suspicion for cancer is, you have to have that piece of tissue to make a definitive diagnosis. And tissue is so precious. And the thing to remember is the minute you separate that tissue from its blood supply, the minute you take that tissue out from the body, that tissue is going to start to degrade. And we want to preserve that tissue as closely as possible so that we know exactly what kind of biomarkers you're expressing on that tissue. So what happens is that as soon as that tissue is removed, we put it into a preservative, a fixative, 10% neutral buffered formalin. We want that tissue to go in there as soon as possible. The tissue is then taken to the pathology lab where it's processed. So tissue is quite soft. If I want to cut tissue and put it onto a slide, I have to make it into a paraffin block. So I make it into a paraffin block. I have a very sharp blade called a microtome, and I make very thin slices of that paraffin block to put that tissue on a slide. And then I can stain that tissue with a variety of different stains that can then be interpreted by the pathologist. But I really cannot impress upon you enough how precious tissue is, and the handling of tissue is so critical so that we can make the right treatment decisions. Okay, so now let's talk about H&E staining, because H&E staining is the first stain that every piece of tissue that comes into the lab gets. And the H stands for hematoxylin, and the E stands for eosin. And so this is a two-dye process. What happens is that hematoxylin is a bluish-purple dye. It stains the nucleus blue, and eosin stains the cytoplasm a pinkish color. Now, I will tell you that the H&E stain has been around since the late 1800s, since the days of Rudolf Virchow, who we call the father of modern pathology, although it's been a while. But this is probably the only test that is as medically relevant today as it was over 140 years ago. Every single piece of tissue is going to get the H&E test. And it's a very important test. We can tell a lot from the H&E. So let me ground you in the H&E. So here we see the cell. You can see the blue part, which is the nucleus, the dark blue part, right, over here. That's the nucleus. And then the cytoplasm is that pinkish color. All right? So that's what the H&E is going to tell us. And we can look at the cell type, the cell size, the patterns, the distribution of the cells, the number of cells. Is there anything abnormal about the shape of that cell? Is there anything abnormal that tells us about any abnormal functions of the cell? So let's give you some examples of some normal tissues. Now don't worry, there will not be a quiz at the end of this. It's really just to be able to show you the different patterns in normal tissue, because we have to know normal if we're going to be able to identify what's abnormal. All right, so let's start off with normal breast tissue. So this is very typical. What we see here, these big white spaces, those are fat cells, because breast is a very fatty tissue. The breast also has ducts to make milk, right? And so these are ducts, and we have lobules. So that's a very typical pattern for what we see with the breast. On the other hand, let's look at an example of normal lung. So here we see this very delicate honeycomb appearance of the lung, because our lung is filled with air spaces for the exchange of gases. We breathe with our lungs, right? So that's the normal pattern with lungs. Here we have colon. So over here, those finger-like projections, those are the glands in the colon. We actually call them the crypts of Lieberkühn. And when we see the crypts of Lieberkühn, we know that we are in the colon. And here we have the kidney. So these round balls, those are the glomeruli. Those are the filtering units of the kidney. Oh, sorry. That's my timer telling me I have to speed up. Okay. So that's what we see. Are we okay with time, Amanda? All right, good. All right, so that's what the normal looks like, all right? Okay, so now let's talk about what abnormal looks like. And we're going to look at lung cancer through the eyes of the pathologist. So I am going to show some pictures of what lung cancer looks like. So if you want to look down, you can do that. Or if you want to continue looking, that's great. But I think it's really important for us to do this deep dive. So what we see here is a typical segmental resection specimen of a lung. Now, most patients with lung cancer will present with advanced stage disease or metastatic disease, where surgery is not an option. So in those cases, we really rely on small biopsies and small amounts of tissue to make the diagnosis. But in early stage disease, surgery does still play a role. And so when we get samples like this, we can actually learn a lot about lung cancer because we have a lot more tissue that we can learn from. So this is our segmental resection specimen. This is the tumor, that pale yellow tissue in the middle. So the first thing a pathologist does is he looks at the tumor with his naked eye, or her naked eye. And we look at the size of the tumor, and we'll see, is that tumor extending to the pleural surface, the surface of the lung? We look at the relationship to the bronchi and the blood vessels. And we can go closer over here, and we can see that that's a fairly well-circumscribed tumor. But we really have to look under the microscope to identify what kind of lung cancer we have. And so if you go under, this is a low magnification microscopic view. So let's orientate you. Over here, you see that delicate honeycomb appearance? That's normal lung. But this here is not normal. That's the tumor. That dense tissue is the tumor. So now we're going to go under a higher magnification to really understand what these cells are that we're looking at. All right, now let me orientate you. The tumor cells are these large cells in the middle. Can you see that okay? Tim, you can see that at the back? Good. All right. So these cells have kind of got a paler pink, almost a foamy appearance because this is an adenocarcinoma. It's a gland-forming cancer, and it makes a substance called mucin. So you know mucus, that kind of sticky substance, is composed of mucin, right? So mucin is what's being made in these cells over here. These smaller cells, those smaller dark dots, those are inflammatory cells. So those are lymphocytes that are surrounding that tumor. Now, I want to hone in on one of these cancer cells, and I'm actually going to point it out with that arrow. So this cell over here. So what's happening in that cell is that mucin is actually pushing that nucleus to the side of the cell. So what you can see here, that's the nucleus, and we call that a signet ring cell. And when we see signet ring cells, I have a very high suspicion that this is going to be ALK positive, right? And so for those of you that don't know what a signet ring is, back in the old days, before we had emails and Twitter and tweets and Snapchat and all the other communication mechanisms, we would write letters by hand. And if you happened to be richer, you could get like your family crest on your ring, and you get some wax, and you put it at the back of the envelope, and you give it to the messenger, and that would take it to the recipient, and the recipient would know that it was your seal, and it wasn't broken, and that nobody had read the contents of your letter. So signet ring cells, when we see a signet ring cell, we're like, okay, this is an adenocarcinoma. I see signet rings over here. Now, these cells here are very irregular in shape and in their appearance. And so this is a high-grade cancer that we're looking at, a high-grade adenocarcinoma of the lung. The more that the cancer cells look like normal cells, the low-grade the tumor is. Now, even though we have a high suspicion this is going to be ALK, before we're going to go on and look at the ALK test, I want to take a pause here to say, just to remind us what the really traditionally the role of the pathologist was. Before we had all of the biomarkers that we can use, the traditional role of the pathologist was to be able to differentiate small cell lung cancer from non-small cell lung cancer. So I'm going to show you what both of those look like. So this is an example of small cell lung cancer. So you can see lots of these dark dots. Those are nuclei, because those small cells have very little in the way of cytoplasm. So it is, as the name suggests, small cell lung cancer. On the other hand, we have many different types of non-small cell lung cancer that the pathologist has to know about. So here we have squamous cell lung cancer. And I can tell that that's squamous because I have this pink stuff in there, which is called keratin, which is very typical of squamous cell carcinomas. Next to it, I have an adenocarcinoma, and I see this pattern here. It's kind of a micropapillary pattern. Here we have another adenocarcinoma, but it's a different kind of carcinoma. It's called carcinoma in situ because it's following the borders of the alveoli. So those are the alveoli at high magnification, and those are the cancer cells. And here we have a cancer that I really cannot tell what it is. It's undifferentiated. So when it's undifferentiated, I'm going to run a whole set of different tests to help me understand, is it adenocarcinoma or squamous cell carcinoma, because this makes a difference to how I'm going to treat my patient. And so we can do these tests called immunohistochemistry tests that detect different proteins. So my adenocarcinomas express TTF1 and napsin A. And my squamous cell carcinomas don't express those proteins, but they will express CK56 and P40. So I can do these tests to differentiate what my cancer is. So if we go back to the example that I showed you, this is what the TTF1 looked like, which was positive. And TTF1 is a nucleus staining, so you can see it's sort of like the round little dots. And napsin A is a cytoplasmic staining, so you can see the pattern is quite different. But this tells us that we definitely have a patient who has an adenocarcinoma, and I've seen those signet rings, so I'm pretty certain this is going to be ALK positive. And when I do an ALK immunohistochemistry, I see that it is indeed positive. Now we can have different patterns of expression of ALK. So these are 3 different examples of ALK that we can see, but all of these patients are ALK positive because they're expressing that ALK protein. Now, ALK immunohistochemistry wasn't always so easy because at the very beginning, ALK expression in lung is much less than ALK expression in lymphoma. And so the first immunohistochemistry test we had really couldn't detect ALK. So this is what happens with immunohistochemistry. We have an antibody that binds to the protein, and then we have a second antibody that binds to a dye complex. And if that antibody binds to the protein and that binds to that primary antibody, you end up with the brown, okay? And so this is what happened with the first IHC antibody that we tried to look at. So this over here is a patient that's ALK positive lung cancer, and this is ALK negative. And you can see no difference, right? I really can't detect that there's ALK. So what we ended up doing was creating these amplification steps. We added more dye steps to that assay, and you can see what a difference that makes. So that was the first detection, the second detection, and the third detection with the amplification. So the ALK assay is actually a very easy assay to read, and any amount of intense brown staining is positive. But there are other ways of looking at ALK. Some of you might have heard about the Elk Fish Test. So this is a fluorescent test where they make probes for either ends of that Elk gene. And when you have the normal elk, those ends are really close together, and so the one probe is red and the other is green. And all you see here is kind of a bunch of yellow dots because those signals are so close to each other. But when we are ALK positive, those signals are apart. There's a break apart, right? And you can see, hopefully, there's red, there's green. There's red, there's green. So when we see that split signal, we know that we have an ALK positive cancer. So when we really think about bringing a test to become a companion diagnostic, the way that we develop our tests is in parallel with how we develop drugs. So if we think about going from a preclinical phase 1, 2, 3, with drug development, it's a similar kind of process when we develop diagnostics. So we start off by really discovering biomarkers, saying which biomarkers do we think are going to make a difference. And then we make a prototype assay. And in those early trials that we partner with our pharma companies, we'll use that prototype assay to see if it's going to work. And if the proof of concept is a success, we then go on and make what we call an investigational use only assay. And then we will submit all of that data together with the drug data to the health authorities, and that gets approved. And so now we have a number of different tests that we can use for ALK. The FISH test, we have the IHC test, and there's also the Foundation 1 test as well. And now, of course, we have a whole slew of new ALK inhibitors as well. Okay, so that brings me then to the importance of biomarker testing. And I think it's really important that we appreciate how far we've come in terms of our ability to characterize lung cancer. So back in the 1970s, when it was just small cell versus non-small cell, then we could look at non-small cell and divide it into adeno versus squamous. Now we have a whole host of different biomarkers. And here we are in 2019, all right? Look at all of those targets, and these are all the drugs that are either approved or that are in clinical trials. So this is progress. This is tremendously exciting. Now, not every drug that's in a clinical trial is going to make it, right? Because you want to make sure that it's going to be effective. But we're seeing this tremendous explosion of research and medications that are coming through. But what this means is that we're going to have to do more and more tests, right? Because we have more and more options. And so the testing landscape can look really quite complicated, right? So when you think about the tissue that you have, I'm not going to go through each of these in detail, but you can see all the number of slides that you need for every test along the way. And sometimes all we have is very small tissue. In fact, most times all we have are small biopsies, and sometimes even smaller than that. So what we see here are 2 fragments of a lung biopsy that's less than 1 millimeter. And of those fragments, only about 10 to 25% of that is going to be tumor. So what your pathologist does is when they look at that tissue, they have to say, how much tissue do I have, and what's going to be the most rational sequence of tests that we can do. It would be great if we had lots of tissue and we could do everything, but sometimes we don't. So we have to be able to use our best judgment to say, what's the best likely option for this patient? And DNA testing alone is not enough, all right? If you look at this progression, right, from a larva to a cocoon to a butterfly, they all look very different, right? Very different looks and very different functions. But if I did a DNA test, it would be an identical DNA test because it's the same thing, right? So DNA on its own is not enough. DNA can tell us a lot of information, but on its own, it's not enough. And I really like this quote from Professor Kerr: the management of patients with lung cancer is becoming ever more dependent on a knowledge of the pathology of each patient's disease. And each patient's disease is going to be different. And not only is your cancer different, but your immune system is different. And your immune system's response to that cancer is going to be different. So each one of you has to know what your tumor is, right? Really know what that blueprint of your tumor is. Now, biomarker testing has been in guidelines since 2013 for EGFR and ALK, and this was updated in 2018 to include a whole host of additional biomarkers. So how are we doing with biomarker testing? Well, actually not so great, all right? So here's a publication that came out last year that showed that only 62% of patients with non-small cell lung cancer in America are being tested for ALK. We need that number to be 100%. This was data that was presented at ASCO, and in fact, Gina and I talked about this data when we saw this and everybody was talking about this data at ASCO, right? So here we have over 1,000 patients with advanced non-small cell lung cancer. 54% tested for EGFR, 51% tested for ALK, even though these have been in the guidelines since 2013, all right? And look at the number that were tested for PD-L1, 48% tested for PD-L1. Now, some of you that we've been speaking to have said you were tested for PD-L1, and thank goodness you got your ALK test back before your PD-L1 test, because you could then get onto ALK inhibition therapy. So biomarker testing is so important, and we really need to know that if you have lung cancer, you need to get tested, right? You need to get tested at least with what's in the guidelines, and you need to find doctors that are involved with clinical trials so that you know what else is happening in the clinical trial space as well. Because groundbreaking medicines are only meaningful when they reach the right patients, right? And that's what we're here to do, to make sure they get to the right patients. So what is my cancer fingerprint? Everyone's cancer is unique, but we have to know what is my cancer's fingerprint. We are the center of our own knowledge universe. And knowing is so powerful. And from what I can tell from everyone in this room, you are so empowered. You know, you want to know more. And we are going to learn together as we work towards the cure. I'm going to end with this quote from Tom Grogan, who is the founder of Ventana. He himself is a pathologist, and he himself is a recent melanoma survivor. He had advanced melanoma and was actually, about a year ago, was completely clear, so we're really happy. Tom retired, but he still stays very much part of our fabric. He is like the grandfather to us all. He is a personal mentor and is a mentor to many people in our community. Phenomenal man. We will not rest until every cancer patient receives the access to information, therapy, and individualized care that we know is possible. And we are bound and determined to be on this journey with you. So with that, thank you very much. I think I've gone over my time, but I appreciate you listening to me.